Okay, hi everyone. Welcome to the lab 11. And we will continue to talk about questions in homework 6. So in the last lab, we talked about part 1 and part 2, which focus on processing glioblastoma microarray data. And it asks you to, the two parts ask you to identify two subtypes by chemist clustering, and you also perform differential expression analysis to identify differentially expressed and differentially methylated genes. And you also perform survival analysis and co-expression. So starting from part 3, we switch to identifying mutations in tumor samples. So in part 3, question 1 and question 2, we will provide you MAF files that contain the mutations of each tumor compared to the normal DNA in patient blood. So here shows an example of such MAF files. So we will provide a bunch of MAF files. Each corresponds to a tumor sample. And if you read it in, and we are interested in the gene mutation and protein change in a mutation. So you may want to focus on just these two columns. So this file records what gene is mutated in this tumor sample, and in this mutated gene, what is exactly the protein change. So this protein change here, for example, means in this gene, in this sample, at position 701, and alanine is changed to a valine. So for question 1, you will be asked to count the times a gene is mutated, combining the two subtypes. So you will need to read in all these files and count the frequency of each gene mutation across all the files. And in question 2, you need to count the frequency of each protein change, a gene and protein change pair, and either combining the two subtypes and in each subtype separately. So in question 2, you will have to concatenate the two columns, the gene and the protein change yourself, and count the frequency of gene protein change pair in the two top subtypes as a whole and in each subtype separately. And in question 1 and question 2, what we want to see is you report something like a table with one column being the gene name or the gene protein change pair, another column being its frequency, actually the occurrence, the count of its occurrence. And in question 2, we want you to compare the gene protein change pair you identified in each subtype separately, and the mutation that is specifically prevalent in one subtype may be the genetic factor that distinguish the two subtypes. So say you see a gene protein pair, a change pair A that happens when counting the two subtypes as a whole, say like 20 times, and you see that this 20 times actually all come from subtype 1, and this particular mutation may be the driving factor for subtype 1. And this is just a clarification on question 1 and question 2. because the results it asks for is not really clear. So for question 1, you need to print out the table of total counts of just gene mutations combining the two subtypes. So it's a table with gene name and the count. And in question 2, you need to first get the total counts of gene protein change pair combining the two subtypes. And then you do the similar thing, but this time in each subtype separately. So in question 2, we want to see three tables of frequency. And there are some coding tips. You are welcome to use Python if you are familiar with it, but I here recommend you still do it in R and you use the apply-R package for reading in files. Remember, we provide the MA file for each sample separately. So here I present a convenient way to reading all the files together and combine it to a large data frame. So you want to use the dplyr function here. It takes a vector and a function, and basically it will apply this function to each element of the vector, and the results will be stored in a data frame. So you may want to get a vector containing a list of file names for the MAF files, and then you put a function to reading the files. And so in this way, you get a data frame that stores the gene mutations and protein changes across all samples. And to count frequency, I recommend using the dplyr pipeline. So you can give it a data frame and then use this symbol, which means the output of whatever command here is used as the input for the next command. In this way, it saves you a lot of intermediate variables. Say you get a data frame, you want to count the frequency of values in column A. You need to first group by the column, and then you summarize it. You count it using the summarize function, and to count frequency, you use n, and you can store the frequency you got in a new column called count. And you can further arrange the frequency by descending order. So you know the top ones are the most frequent values. And this screenshot shows an example of using this pipeline. So I use the data, the lung disease data from survival package, and I want to know the frequency of values in the ph.column. So I group by it and summarize it and arrange it by descending order. And so I know that, for example, in this column, the value 90 appears 74 times and 80 appears 67 times, and so on. And you can count the gene mutations, the frequency of gene mutations or gene protein change pair in a similar way. And in the next three questions, question 3 to question 5 in part 3, we focus on studying the the tumor driver genes. So this is some background information. So imagine, like, this is an analogy. Imagine like your cell is like this car here, and on the left side is progression to cancer. Normally, you have in your cells proto-oncogenes and tumor suppressor genes. So proto-oncogenes are usually genes that promote cell growth. So it's like the gas for the car. And tumor suppressor gene is genes that stops over proliferation. So it's like brakes. So these two types of gene functioning together will ensure that your cells will proliferate and replicate normally, but it will stop at certain time points. So it does not, the proliferation does not lose control. However, in the cancer setting, there are two possible mechanisms that could lead to cancer. The first is that there could be some gain of function happening in your proto-oncogenes, and it will become an oncogene. And using this analogy, it's like the genes overactivated, like your car having too much gas, so you will progress towards cancer. Another mechanism, you lose the brake. That is, it could be some loss of function mutations happening in a tumor suppressor gene. So you lose those checks on your cell cycle, and without a powerful brake here, you can also progress to, your cells may also progress to cancer. So for question 3, you will ask to identify uh, cancer driver genes in glioblastoma. So you will go to this website, and you will choose subset to glioblastoma. I mean, subset studies that are related to glioblastoma. And then you should choose studies you are interested in, and you should also submit the top genes you get from the question 1. And then you look at the positions of mutations, determine whether a gene is likely to be a driver gene. So here is a sample view of invasive breast carcinoma, and I checked 4 genes. And on the very top, it's each color represents your study of origin, and down here you get the mutation of mutation across these genes. For example, for BRCA1, we see it has a lot of amplification, also have deep deletion, and similar is true for BRCA2. And there is no like quantitative way to determine which gene is likely to be the driver. But if you get multiple genes, you should look at those with many mutations, or they have mutations across a large section of their gene body. So you can identify which ones are likely to be the driver gene. And in question 4, you will be asked to identify whether a mutation is gain of function mutation or loss of function mutation. So you can click into that mutation and see whether it happens frequently at a specific position, or it happens like at very low frequency in across the gene body. So for gain of function mutation is usually a single a single mutation that happens frequently at a very specific position, because for gain of function, you may need to change a very specific protein structure at a certain location, changing a certain, say, amino acid. For loss of function mutation, it could happen at many places of the gene body. So leading to change in many, could be a very random place position in the protein. And this change will very likely lead to loss of function of the protein product. So for loss of function mutation, it may happen across many spots with low frequency. And for question 5, it's very easy. It asks you to look to determine whether patients with a mutation have better survival compared to patients without the survival. So you just need to look at the survival curve, and it will generate for you in this website, and take a look at its log rank test p-values to see if the two survival curves with the mutation or without the mutation are significantly different. And for question 6, it's also very easy. You just it asks you to count the number of clinical trials targeting a certain mutation. So you just go to this clinicaltrials.gov website, search by the disease name, like glioblastoma, and the gene name. And and it will print out the clinical trials targeting this gene in glioblastoma. And there are two ways. You can export a file as a TSV document and read it in R and count using code. Or if, say, there are just 10 trials, maybe you just manually count on the website for those that are still, I think the question asked, they are still recruiting people. So it's going to be very easy.